This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 163, recorded December 23rd, 2011. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today, right across the desk in my office, a infected Dickson Despommier.
At least it feels that way. Who knows? This could be a symptomatology, but I do feel a little bit down and under the weather.
Dickson, would this be your last TWiV?
I hopefully not. It's a volunteer thing anyway, isn't it? Please invite me back.
Dickson has some kind of an infection which is giving him systemic aches, malaise, fever.
Yep.
I'll bet it's flu.
It probably is.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
Are you viremic?
Well, I was just recently. And I'm just kind of getting over it. So this will be kind of a barnyard episode. We'll be talking about chickens, and Dickson and I are a little hoarse.
You cheered him up. That's good.
We'll just have to pony up to all that stuff anyway.
Ooh. Also joining us from North Central Florida, Rich Condit.
Hi, fellas.
Hey, Rich.
How you doing?
Hey, Rich.
All right.
Could be better. Actually, I'm brightening up. So
Dickson, he was moping all afternoon in my presence, and now he's happy.
Be happy, Dickson. All those symptoms mean your immune system is working.
That's what Vince tells me.
Yeah, the fever, the aches.
That's right.
All that.
Yeah.
That's
interferon, dude.
Yep.
Listen,
Very good shape.
I'm on point for that. Believe me.
Okay, good. You'll be okay, Dickson.
Yep.
I don't think you're going to die.
No, not
if
right.
that's what you're afraid of.
No, of course
But
not.
don't fear death. You are 70.
I'm not. I'm 71.
Is that
Could
bad?
be any minute.
I get this every day when I come to work.
From who?
From you.
I do. Sorry. All right. What's everyone doing for the next few days? Rich, you got people at your house, right?
Yeah, we got most of my family around, and it's just a gas. I'm up to my eyeballs in grandchildren.
Cool. Have
Very,
a good time.
Very cool.
Yeah.
What are you going somewhere, Alan?
Yeah. I'm gonna
Go down to Westchester.
We generally spend Christmas with my in-laws. They're right outside New York City.
No, be careful.
Cool.
The roads,
Yeah.
driving.
Yeah.
Exactly.
Excellent. And
How about you?
Dixon's not doing anything, right?
No, that's not true. As soon as I recover, and that should be by tomorrow,
Yeah, if you think so.
we are remodeling a major portion of our co-op. So,
Cool.
we're having a good time doing it, but it's been stressful.
Nice.
And how about you, Vincent?
Yeah,
I'm
Vince.
staying home. I'm writing grants.
Great.
No, we have 3 kids, of course, and we're staying home. We're going to enjoy the holidays. But Rich, I just finished an R01.
Fantastic.
For those listeners, that's a grant application. And I have 2 or 3 others that I want to write. So I'm thinking about those. But I'll be around next week.
All right.
Next week, we're also doing a TWiV. By the way, on the 30th, I think that's next Friday.
TWiP.
Yep, 7 days from now, we're going to review 10 cool stories of 2011.
Very nice.
That's right. Time
Very
to do
nice.
that.
And there are a couple that, we're going to review.
Yeah.
So you might want to tune in for that. Before we start today, take the listener survey, please, at triplemojo.com/TWiV. For those of you who have not, of course, and those of you who have, we thank you very much for participating in that. It's almost
thank
there.
you.
Also, go over to the ASM site and post your idea for a scientific session. There are plenty of good ideas running around that you can suggest for a session. If you'd like to do something like that, we'll put a link to that in the show notes, as usual.
We are approaching our goal of 1,000 on the listener survey, only asymptotically. Right?
We will actually never get there,
right?
but it's
so
close.
give us a big Christmas present, okay? You're all on break.
Right.
You're taking, some time off. Give us a couple of minutes here. Give us a Christmas present.
Right.
Fill out the listener survey.
That's right. Or a Hanukkah gift.
Thank you.
Right. And remember that subliminal message from last time. If you're driving while you're doing this, next time you're at a computer, you will remember. TripleMojo.com/TWiV.
Stroke, nice. You're British. Okay, today we thought we would discuss a paper which was published in 1911.
Wow.
That would be 100 years ago this year.
It's even before my time.
And that's
Barely
why,
got in under the wire.
Yeah, we figured we had to do this year because it's the 100th anniversary of the publication of a paper by Peyton Rous. He's the only author on the paper.
Interesting.
It was published in the Journal of Experimental Medicine, and its title is A Sarcoma of the Fowl, Transmissible by an Agent Separable from the Tumor Cells. And it was received for publication on February 9th, 1911, 100 years ago. So this is quite a paper for reasons we'll go over today. 1911.
Yeah.
Now, in terms of virology history, by 1911, of course, viruses had already been discovered. The agent of tobacco mosaic had been discovered before 1900, of course. Foot and mouth disease virus was 1898. Yellow fever virus, 1901. Rabies, 1903. Variola, 1906. And polio, 1908.
At this point, however, people didn't know what they were, right? They knew that they were
small
agents that were submicroscopic.
Right. And they went through a filter that would hold back bacteria.
Right.
And so
they could be propagated in a fashion that required living cells or tissue to propagate them, and they appeared to grow. Indeed. So it wasn't just a chemical.
Right.
So virology at this point in history was just revving up.
It was.
We'd found some viruses and had an inkling of what was going on, but not really a whole lot of information.
Now, actually, I was thinking that the electron microscope wasn't invented until the 1930s.
Correct.
Right.
But vaccinia virus, pox viruses, they were actually able to barely see with a light microscope before that, but I don't know what the date on that was. So at any rate, sometime around before the 30s, around this time, one of the viruses at least could be associated with a particle of some sort.
Mm-hmm. That was an important advance later on when people could see that viruses were particulate, right?
The electron microscope was a big deal.
Yeah.
You bet.
For a lot of reasons.
Well, yeah, still is.
Yeah.
Now, this paper, actually, to understand it, we have to refer to the previous paper by the same author, Peyton Rous, who was an MD working at the Rockefeller Institute for Medical Research. That's what it was called back then.
Yep.
When you went, Dickson, it was called
Rockefeller University.
Yeah.
In fact, they had just closed the Princeton campus for the Rockefeller Medical Research Institute.
So you were there in 1912, right?
13. Please, let's not.
Shortly before the Great War.
Exactly.
Was that soccer, Alan?
Yes.
No, I went to Rockefeller in 1967.
All right, so we'll have your Rouse stories today for
Yeah,
sure.
I've
That's
got them.
why I'm happy you're here.
Oh, me too.
The paper the year before was entitled A Transmissible Avian Neoplasm, and in parentheses, Sarcoma of the Common Fowl. It was also by Peyton Rous. He's the sole author on this paper, both of them. Just him.
That's actually pretty common in the old literature.
Here.
In the really old literature, yeah, because the experiments, were things that an individual could do, and I think Rous had originally gotten a $200 grant to pursue this particular chicken too.
So we have, as sort of background, there's a wonderful book which we have mentioned as a pick before. It's called The Emperor of All Maladies. It's a book about the history of cancer,
right,
by Siddhartha Mukherjee, who happens to be here at Columbia, Dickson. Did you know that?
I did, yes, I did.
He's an oncologist. And he writes, in 1909, then a 30-year-old scientist freshly launching his lab at the Rockefeller Institute, Peyton Rous had been brought a tumor growing on the back of a hen of a black and white species of chicken called Plymouth Rock.
Yep.
A rare tumor in a chicken might have left others unimpressed, but the indefatigable Rous secured a $200 grant to study the chicken cancer. Soon he had categorized the tumor as a sarcoma a cancer of connective tissues with sheet upon sheet of rhomboid foxide cells invading the tendons and muscle.
Cool.
That's why this book is so cool. It's really written very well.
I've read the book, but this selection you sent, rereading it, sort of recharged me on this. This is one of the most outstanding books on science I've ever read. It's really good.
Oh, wow.
So in this initial paper in 1910, he basically reports that he got this chicken, this Plymouth Rock chicken with a big tumor on it, and he was able to transplant the tumor into another chicken.
That's not a novel finding. Okay, people had been able to transplant tumors of one sort or another, though this was the first time in chickens, right?
Sure.
Yeah, so actually in the beginning he talks about some of the other transplantable tumors, and he also mentions this experiment from 1908 by Ellerman and Bang, who showed that chicken leukemia was transmissible. And he actually says they have shown that the disease is dependent on a filtrable virus. I think that's the only time he mentions virus in both papers.
That's right. He's very conservative in his own conclusions.
So, but the thing that I've understood from reading some background is that people didn't think leukemia was really a cancer.
Right.
Right?
Yeah.
Right.
Because I guess the idea was that tumors were solid things, right?
Exactly.
Right.
Well,
so interestingly, in Mukherjee's book, the first cancer that he describes is a lymphoma.
Yeah.
Okay? It wasn't recognized as a cancer at the time, but it was one of the first diseases that we now recognize as cancer that was pursued vigorously.
Right.
Yeah, and leukemias, they just seem conceptually a little different because you don't have a tumor or a mass you can point to, and it's a blood disease, and so it's
Sure.
So this is very funny in this paper. These papers, as you guys did the Tort paper earlier.
The Tort paper, which by the way also had just a single author.
yes,
It's just very conversational.
Mm-hmm.
yeah.
It's nothing like the papers of today.
The sort of methods read like it's transcribed directly from his notebook.
So
Right.
It's just funny.
Like a
It's
log.
like the tumor here reported was found in a barred Plymouth Rock hen of light color and pure blood. What does barred mean, Dickson?
Barred, this is stripes.
Stripes?
Yeah.
It had existed for some 2 months before the fowl was brought to the laboratory, becoming noticeable when it was 15 months old.
Do they say where the chicken's from?
From other sources, I gather it was a Long Island farm.
I've heard it's from Long Island. Can you imagine someone bringing him this chicken?
I actually heard it was from Jersey, but that's...
It would be like someone bringing me a tube of their blood, I guess. Which I would be
Or a
illegal.
mouse.
Well, at that
Right.
time, New York City was surrounded by farmland,
Sure.
so that you would take it into the city to see
That's
what
right.
was wrong with it, I guess.
Right, and there were very few places to take it to.
Yeah.
So he describes the tumor. It's a mass projecting from the right breast. It developed slowly. The chicken was okay, and then he sliced it open.
Right.
And then the chicken wasn't so okay.
So he said it looked like a sarcoma. Guys, what's a sarcoma? Can we tell the people out there?
Right. So
That's a tumor that originates from mesodermal tissue.
Right.
And what's mesodermal tissue?
That is one of the 3 germ layers in an embryo. You have it's between the endoderm
And the ectoderm. That's right.
And so it would include tumors of bone, cartilage, fat, muscle, vascular, and hematopoietic tissues.
Correct.
Right, and the ectoderm makes your skin and also your brain. And the endoderm is going to make blood cells.
All right. So sarcomas are solid tumors, right?
They are.
And sort of the counterpoint to sarcomas or tissues or cancers of the mesoderm are carcinomas, which originate from epithelial cells. And so you get malignancies of tissues such as breast, colon, and lung.
Right.
The other thing to note here is that if this chicken had been brought to Peyton Rous several years earlier, he might not have had the technologies necessary to actually preserve it, embed it in paraffin, cut sections, and stain it.
Is that right?
Yeah. Because
Really?
the Romanovsky stands, if you go back, I actually had the privilege of visiting the Pasteur Institute one year, and Mechnikov and Romanovsky have portraits right next to each other because without the ability to look under the microscope, you're not going to see anything. So that's how he could describe these tumors. And I actually knew another guy there. His name was John Nelson, and he was an old-time pathologist. And he made his entire living describing pathological conditions under the microscope, even well into the 1960s and early 1970s.
So he says he took bits of this tumor, bits, that's
Right.
a quantitative.
Yes.
Bits of it were all at once inoculated by means of a trocar into the other breast and peritoneal cavity of the host. So he just re-inoculated the same. What's a trocar, Dickson?
It's like a tube with a point on it.
Well, it's a hypodermic needle that has pores on it to allow thick, viscous fluids to move through it.
So the stuff goes out the sides of the needle as well as the end.
Yeah, some of them are. I don't think they're all configured that way, but I think most.
And they also put these bits into two hens from the same setting of eggs.
Uh-huh.
You mean they were? What does that mean? The same setting?
Well,
in
the same
1911,
hen.
everybody was familiar with agricultural terminology,
That's
but
right.
it's the same litter, essentially.
Is that true, Alan? Really, that everyone knew what he was talking about, right?
Probably.
That's interesting.
You see, I've read a fair amount of the old literature just going through my grandfather's papers, and you go back to the 19-teens and you see references to farming terminology from the time, and it's just that's part of the scientific literature because that's what you were working on.
That's right.
Wow.
When he says the same setting of eggs, I'm not sure that he says later on that he doesn't know the exact genealogy of these guys because I'm guessing that this is like a hen house or something.
Yeah.
Right.
So by the same setting, they went in on the same
same
day and collected these eggs.
Right.
Okay.
Right. I shouldn't have said the same litter. It was probably a bunch of hens in the hen house, and the setting would have been that day's eggs.
And that's relevant because he himself bemoans the fact that he doesn't know the precise genealogy. Becomes relevant.
Yep.
35 days later, the original host was dead of intraperitoneal growths.
Oof.
Dead because he sliced up the tumor, so obviously he did it properly. And then there was a nodule in the breast of one of the other chickens that he had put in these bits of the sarcoma.
And by the way, for listeners who are wondering about these papers, these are free online. The National Library of Medicine has them. We'll have links in the show, so you can actually go and read these papers, and they're really cool. Yeah,
I would recommend it if for anything. It's a historical couple of papers, and also it's very different. As you can see, we're just amazed at the terminology because it's totally different from today. It's quite easy to read.
Yeah, it's exactly
quite easy to read, right.
It's a great thing I know a lot of members of our audience are not in virology or they find some of the terminology confusing. If you go back to these old papers, things were a lot simpler back then, and you can see where this stuff came from.
So he goes on to discuss in some detail the inoculation, which we just talked about, and he talks about the tumor. He looks at sections under the microscope and characterizes it a little further. And then he says, following the growth's successful transplantation, an attempt was made to propagate it further. And then he says it's now in the 4th generation. All right, so he's able to pass it 4 times from chicken to chicken. And then he says the tumor has never thriven except in the intimately related fowls of the pure blood stock in which it was first noted.
Uh-huh.
You like that, Dickson?
I do.
What does that mean?
These are inbred strains of chickens, and the virus didn't jump between that strain, let's say. You wouldn't call it a species, but it was certainly an inbred strain.
So my reading is that the cells are probably rejected in a different histocompatibility.
Right. At this point, he hasn't made a cell-free thing. So we don't have any evidence at this point that he's dealing with a virus at this point.
Right.
Correct.
He's transmitting tumor cells.
Yeah.
And there's probably a, histocompatibility thing going on.
Yeah, he's putting cells in. I bet they're just rejected if you use a different line of fowls, which we didn't know about back then, of course. So that's kind of cool. He says the transplantation appears to succeed better in young fowls, which probably because they don't have as good an immune system, I would guess, right?
Right.
Bursa Fabricious probably hasn't developed fully at this point.
What is the Bursa of Fabricious,
It's a
Dickson?
thymus-like organ.
Why is it called the Bursa of Fabricious?
Well, because of Fabricious called it a Bursa, and so from that point on, it has always been known as that.
Okay.
It's too bad they didn't, if they'd have carried on a tradition like that, you could have called the polio receptor the receptor of Racaniello.
Exactly.
Yes.
Right?
And many of us do. In fact, we're forced to every time I come to work.
I would never, ever do that.
You would never do that?
I would never impose my name upon the scientific establishment. No.
I don't know. Racaniello has quite a ring to it.
It does.
Ooh,
No,
okay.
it's nice.
But I think you should name things so that they make sense, not after people.
Right.
Yeah, like E2F and
Exactly.
NF kappa B.
Sounds great.
Okay.
He goes to say that the tumor is at best so difficult of propagation that no attempts have been made to determine whether it can be transmitted by cell fragments or cell-free derivatives. So he's having trouble passing it from bird to bird. Mm-hmm. He only has $200. What do you expect?
Exactly. But in those days,
I just did a quick the inflation calculator, which unfortunately the inflation calculator only goes back to 1913. But
darn,
$200 would have been about 4,500, maybe closer to 5,000.
not bad.
You know,
So it's not a lot of money.
Wouldn't
No.
go far today. You can buy a few kits with that, and that's it.
Yeah.
Right.
Let's see, what else should we do from this paper? So that's basically it. The first avian tumor proved transplantable to other individuals, right? And here's at the end they have a picture of this this barred hen with the tumor
That's right.
shown here.
If David Baltimore is listening, I'm sure that David is fully aware of the fact that fly fishing depends on these inbred strains of chickens in order to produce feathers of different colors and types for creating models of aquatic insects. And so this was a big business in those days, by the way. Fly fishing had emerged into the light of day from England. This guy, Theodore Gordon, was up in the Catskills inventing this new way of fishing, in this country at least. And they had to raise chickens in order to make this thing happen. And so these guys weren't just raising chickens for food, they were raising them for recreation as well. How about that?
So
But don't get your hackles up about it.
going back to Mukherjee, just to sort of summarize this, he says in 1910, unwittingly Rous threw the somatic theory of cancer. So this was a somatic mutation hypothesis that environmental carcinogens somehow altered the cell and caused cancer.
Which, of course, it can, but it took a long time for that to actually be proven, yes.
So he threw this into doubt by injecting this tumor from one chicken to another and showing it could be transmitted. All right, so that's not consistent with this somatic mutation hypothesis. And so Mukherjee says this was curious, but nonetheless still understandable. Cancer was a disease of cellular origin, and transferring cells from one organism to another might have expected to transmit the tumor. But then Rous stumbled on an even more peculiar result, and that's the next paper, which is the real one, the 1911. A sarcoma of the fowl transmissible by an agent separable from the tumor cells. So what he does, he grinds the tumor up in sand.
Right. And resuspends it in a quantity of a considerable bulk of Ringer's solution.
Oh, man, what would the science editors think about that?
Well,
And
Well,
shaken for 20 minutes in a
I don't know
machine.
I don't know if they still do this, but for the first several years that I made extracts from E. coli,
yes.
some of you guys have probably done this. Yeah, I'm sure you have. We used, to break them up, we used a mortar and pestle
Of course.
and alumina, right? Which was, I guess, an abrasive.
Sure.
And you ground them up in a mortar and pestle.
Yeah.
So that's only slightly more sophisticated than this.
Well, and people still douse their cells, right?
Yeah, and you've always got the French press standing by in case everything else fails.
And I love it. It was separated. The sand and tumor fragments were separated out by centrifugalization.
Yes, centrifugalization. That's so funny.
In large tubes.
Indeed. Indeed.
For 5 minutes at 2,800 revs per minute. I'm wondering what kind of a machine that was.
Right.
It's maybe hand cranked.
I was wondering.
It's a graduate student out in the field
crank thing.
twirling this string around with a big tube at the end.
So they take this fluid, which so he passed some of it through a filter. All right.
Yep.
Now he starts the paper off by saying ordinary filter paper
I was
was
going to
used.
say which filter. Ordinary filter paper.
Initially, he said it was enough to hold back everything except a few red blood cells and lymphocytes, but he said it would hold back the tumor. But then he goes on in his methods to say he used a Birkfeld filter, a number 2 course, and he filters the tumor extract through this, and then he puts it into chickens and he gets tumors. Of 22 receiving both filtrated tumor, 5 develop sarcoma. And then he says, well, these filters let bacteria through, so let's do it again. He uses a number 5 filter, which he says holds back the bacteria.
Right. Specifically, he's looking at Bacillus prodigiosus, which has since been renamed to Serratia marcescens.
Ah, yes.
And I'm assuming he looks at that because it's easy to see, right?
It's kind of a pinky color, right?
Yeah, I suppose easy to grow or whatever.
Right. You've done a TWiV episode on Serratia, haven't you? Or have you?
No, we haven't, no.
Oh, that'd be a good one.
Yep.
You say Serratia, I say Serratia.
Oh, okay.
I think it's...
Serratia?
Sure. I think it's Serratia.
Okay.
That's what the Army calls it.
Well, I'll go with that then. All right.
Well, anyway, the first shot of this number 5, it doesn't work. None of the chickens get tumors, and then he does some business with heating it, and which didn't make any sense to me. But eventually he gets it to work. He gets tumors in chickens after passing it through this number 5 filter. All right. So
yeah, it's interesting. It's like he tried keeping it at somewhere around the body temperature of the fowl, I guess.
Right.
Thinking maybe that deviating from that he must have been thinking it was alive.
Mm-hmm.
Right. Deviating from that would kill it or something?
Could be.
Not
I mean,
clear,
nowadays, we keep everything cold.
Right.
Yeah.
Right.
Yep.
So basically he gets tumors from this filtrate, which doesn't have any cells in it as far as he can tell. And he says if you take those tumors, you can then pass them on to other chickens as well.
You mean other strains of chickens in addition?
No, the same strain, I think, mainly.
Mm-hmm.
Well, initially, yes.
Initially.
Most of the paper is a lot of histology looking at this tumor.
Lots of histology.
And transplanting it.
Yep.
Showing how it feels.
Should we pause? Should we pause at that moment, Vince?
Yes.
Should I mention to the group that I've actually seen that slide?
Oh, yeah, definitely.
Yeah, well, I was there in the 60s.
I thought you did the experiment.
No. But I was asked one day, would you like to see the original histological section from the chicken that launched the concept of viruses as tumor agents? I said, sure. Who would say no to that? And the guy took me over to this big cabinet with histological slides in it and reached way in the back and pulled out a sleeve of slides, and he held it up to the light, and he says, there it is. I was stunned.
What did it look like?
It looked like a large pink and blue tissue underneath a cover slip. And they used to mount them in balsam in those days because they didn't have the synthetic mounting media that we now have. So they used the resin from the balsam pine tree.
Mm-hmm.
Yep.
And you could smell it. You put it up to your nose and you could smell the balsam.
So they're still there, I presume.
I hope so.
Yeah.
It was quite a remarkable historical moment for me, at least.
So you met him, right?
I met Peyton Rous, yes.
And by that time he was
set to retire.
Old guy.
That was the year he got his Nobel Prize. So I think I'm correct in remembering that. 1967. Somebody please correct my failing memory if that wasn't the year.
It was 66 or 67, I think, was his Nobel Prize.
Yeah. And ironically, he had alienated so many people.
66, yeah.
66. Okay. That was the year before I came. And he had alienated so many people in his county in Connecticut that the Connecticut Medical Society had refused him membership. And then when he won his Nobel Prize, they couldn't hold out anymore, and they elected him a member of the Connecticut Medical Society. So some intrigue in politics and everything.
So summarize him in a few sentences.
Well, I mean, he was frail. His best friend was Eugene Opie, and Eugene Opie was still alive, too. In fact, he outlived Peyton Rous. Peyton Rous died of pancreatic cancer. And so I got to meet Peyton Rous, and his laboratory was down the hall from Bill Traeger's lab, who did a lot of work on malaria, for instance, and termites and all kinds of other interesting symbiotic relationships. In fact, Bill Traeger became famous for having been the first person to culture malaria in vitro. But he was a good friend of Rous's, of course, and they used to talk at lunch a lot, and Rous had, by that time, become a bit of a recluse. He had been vindicated with regards...
a spider?
No, stop it. You know too much, Vincent. But that was a good reference. It was hard to get to know, but Eugene Opie was really easy to get to know. He would talk to anybody. And Eugene Opie was the guy who actually discovered the role of the gallbladder. Imagine, if you go back in biology so far, nobody knew what the gallbladder was. He said, hell with it. Let's just take it out and see what happens.
So you used a word to describe Rous today at lunch.
Yeah.
What was that word?
Curmudgeon-y?
Yes, there you go.
Yeah, he was a bit of a curmudgeon because he knew he was right from the start. And he was dealing with the politics of medical science in Connecticut. And he was not recognized fully by that group. But at the same time, he rose to the level of editor-in-chief for the Journal of Experimental Medicine. And one of his dear colleagues, who became a dear, dear friend, was Charlotte Friend, who discovered mouse memory tumor virus. And the moment that she said, I have another virus that causes tumors, he published every one of her papers, and he used to reject papers that actually couldn't replicate results and get definitive results and say, oh yeah, it works in my lab too. A lot of people couldn't get that sort of thing to work. So Rous became a bit of an angry scientist at one point.
Well, it was over 50 years between the original discovery and the award of the Nobel Prize, which is something close to a record, I think.
Yeah.
Correct.
It is.
They could no longer withhold it.
Back to the paper, he says, the tumors which result from the injection of a cell-free filtrate take much longer to appear. Interesting, right?
Yes.
So if you put cells in, it's faster than putting in the cell-free, which I suppose makes sense.
Makes sense to me.
Right?
You know, I see
That's right.
having to do an infection, and the cells are probably already going.
But they didn't know how many virus particles either, so he was just guessing.
Right. He actually doesn't mention virus in this paper. Here's another cool sentence. A fowl which has once failed to develop a tumor usually proves resistant on later inoculation.
Refractal.
Yes. That's very interesting.
Yeah.
Of course.
And he also points out, does he not, that sometimes a tumor starts to take and then regresses, and that fowl is subsequently resistant.
Yeah.
Is that correct?
Yeah.
Did I read that correctly?
Sure.
Yes, and he also saw that some of the chickens got sick, and when they intercurrent illness of the host may check the further development of a nodule 2 or 3 centimeters in diameter and may even cause its retrogression.
Sure.
Yeah. So how do you interpret that?
I think it's the secondary infection has triggered an immune response that then noticed the tumor.
Right. That's my read on that too. It's fascinating.
I can show you lots of. Well, we know that BCG does it, and I can tell you that Racaniello does it too. So there's a lot of general lifting of the immune system above a certain subclinical level that actually protects you against all kinds of things.
Right. And he has this chart too that shows the blobs of different sizes showing the size and shape of each tumor.
No, you didn't know what was important and what wasn't, so you have to keep track of everything to begin with, right?
Yeah.
But this is funny data. He draws the tumor with a pen or something.
By the way, this is where
no Photoshop.
I was going to say this is where Photoshop got their ideas. I must also mention that there was another group working on tumors at Rockefeller that were absolutely spot on in their work, and Rous used them all the time as checkpoints for the correctness of the hypothesis that viruses do cause tumors, and that was Armin Braun, B-R-A-U-N, Armin Braun, and he was studying plant tumors. You know these galls on trees that you see?
Sure.
Right.
That's a tumor, and you can transmit this thing from one tree to the next. It doesn't even matter which species of trees. So he was busy trying to watch them regress. And Armin Braun was the first person to suggest that tumors regressed. He was the official recipient of that piece of knowledge. Everybody said, oh yeah, it does it in plants. Maybe it does it in animals too. So they had a lively group there. He wasn't in the same building that Peyton Rous was, but they used to talk a lot.
So he also says with repeated transmission, the rate of growth as well as the percentage of successful transplantations has increased in the period which elapses between implantation and the appearance of the new sarcoma has reduced from 4 weeks to 4 or 5 days.
Wow.
Right, so as he keeps passaging it, he's selecting for faster growth, presumably.
Right. Or giving more particles. You don't. Maybe he got better at it. As he got better at it,
Then he talks about metastases because these tumors spread within the chicken. And he says this is probably because cells are moving around, and I think that's that's likely to
Did
be
he
the case.
do any serial dilution experiments to show what the minimum dose was?
No, he didn't do it in this paper, no.
Interesting.
I mean, he really doesn't focus on this being a virus. He just says it's filterable.
Well,
right? He never really gets to this. And I don't know, he says in the end, he calls it a minute parasitic organism.
Right, he says that the tendency will be to regard the self-perpetuating agent as a minute parasitic organism.
That's really hedging.
And then he goes on, but an agency of another sort is not out of the question. Could be a chemical stimulant, et cetera, et cetera.
Sounds like the caveat at the end of the Watson and Crick paper.
Right.
I mean, I'm just wondering, and that's really the last bit of that paper. Much of it is just characterizing the transplanting the tumor itself.
Sure.
When does he say, in this tube I have a virus that causes cancer? I don't know when that happened, actually. It took many more years probably after this.
That's correct.
Of course,
but this sort of genealogy of the passages, chart 1, is also kind of interesting because as he goes through subsequent passages, he eventually does get it to infect and produce tumors in more distantly related chickens.
Mm-hmm.
Right. So to me, this is one of the most interesting things about the paper is that with passage, this thing becomes
a more general,
basically more virulent.
Right.
Okay.
Right. Thank God he didn't stick himself.
And good thing he wasn't working with ferrets.
Right, which is the universal host for everything, of course. Can somebody check to see when Charlotte Friend published her first paper on the mouse memory tumor virus?
If he had done this in ferrets, he wouldn't be allowed to publish it.
That's right.
You want to know when Charlotte Friend published her first paper on viruses?
I do.
Friend.
It's probably in the 20s.
I spelled virus wrong. Okay.
See, because that was the Sentinel paper that gave Rous confidence to go forward. So I'm told.
Really?
Yep.
Mm-hmm. Okay, 1950s.
Yep. It was a long time.
1957 is a citation I found.
Right. So up to that point, no one had shown that what's really in the filterable solution is a virus. And the moment she did that, it was a validation
Let's
of his research.
see what Mukherjee says.
Yeah. Well, that's the story that I got while I was there as a postdoc.
So he says, remember, I left off by saying he stumbled onto an even more peculiar result, shuttling tumors from one bird to another. He began to pass the cells through a set of filters, which we've talked about.
Right.
He expected the tumor transmission to stop, but instead the tumors continued propagating. I didn't get that from reading the paper, actually. Did any of you?
No.
So what was that again?
By filtering, he expected the tumor transmission to stop. I didn't get that from the paper at all.
I didn't see him mention that.
The agent responsible, Rous concluded, was not a cell or an carcinogen, but a tiny particle. As we've read, he didn't really conclude that. He sort of hedged his bets.
Why couldn't it have been a chemical?
Yeah, he considered that also.
Right, and he says at the end that he hasn't picked a hypothesis here.
Exactly.
We've not adopted either hypothesis.
Right.
So Mukherjee says basically his virus was later called Rous sarcoma virus, RSV, but I don't know when actually that happened. Now here's something for you, Dickson.
Uh-oh.
He says in 1935, a colleague of Rous's named Richard Shope reported that a papillomavirus caused tumors in rabbits.
That's right. He had the lab next to Rous's.
And I understand that Rous encouraged Shope to look into this.
Absolutely.
Which is, the two people in the same place. It makes
Sure.
sense. 10 years later, in the mid-40s, a leukemia-causing virus in mice, and then in cats, but not humans, of course. 1958, Burkitt's lymphoma.
Right.
First human tumor virus.
So take a guess at what
Richard
Shope and Peyton Rous died from. I already told you that Peyton Rous died from pancreatic cancer.
Mm-hmm.
So did Shope.
Yeah, it's probably the bad stuff they worked with without gloves and masks.
Yeah.
Now.
In the next door, the same laboratories. Nobody wanted to go down there after a while.
Yeah, we didn't have any environmental health and safety departments back then to tell us not to mouth pipette.
That's right.
I don't know. This is just anecdotal, but I know a lot of scientists who've died of pancreatic cancer.
I do, too.
I mean, Bernie Fields and a guy who used to work here. I don't know why. Someone should do some epidemiology.
Someone should get the denominator. That's the thing.
Yeah, exactly.
They've actually done that, but it doesn't give you the answer you want.
There have been, I know that there have been at least, I have some colleagues who died of, who worked on SV40, died of colon cancer, and there was a lot of effort put in to try and figure out whether SV40 was associated with that, and they came up with negative results.
Yeah. Now, basically, because of EBV, Epstein-Barr virus, in 1958, this rejuvenated the whole idea that viruses cause cancer in people. And Mukherjee writes, a Life magazine cover in 1962 said, cancer may be infectious. And so he says, Peyton Rous was rehabilitated into the scientific mainstream and levitated into permanent scientific sainthood. As a consequence. So all these years, nobody found any human tumor viruses. And when they finally do, they say, Peyton, that's why you got his Nobel Prize.
And the other thing, of course, was because he was the editor-in-chief of the Journal of Experimental Medicine, and people publishing articles contrary to his own hypotheses often didn't, in their view, get a very fair review of their papers. Yeah. So he was controlling the information as well.
Well, but even then there were other journals.
Oh, but of course, but that was the highest one at the time to come out and reach all of the cognoscenti with regards to viruses and tumors.
Right. It's worth reading a few of these Mukherjee passages here. On the evening of December 10th at the ceremony in Stockholm for his Nobel Prize, he rose to the podium like a resurrected messiah. He acknowledged that the virus theory of cancer still needed much more work and clarity. What have been the fruits of this somatic mutation hypothesis? Most serious of all, the results of the somatic mutation hypothesis has been its effect on research workers. It acts as a tranquilizer on those who believe it. Can you imagine saying that?
Yeah, well...
the appeal of studying Rous virus was further compounded by the formidable force of his personality. Bulldogish, persuasive, and inflexible, he had acquired a near paternal attachment to his virus and was unwilling to capitulate to any other theory of cause, which is not good.
No, it isn't.
No. But science is done by people. That's why, as you say, Vincent, you should trust science, not scientists.
But the guy who worked for Rous that I knew was an Englishman, and eventually went up to the McArdle Cancer Center, and I'm blocking on where that is, but you guys know where that is. Wisconsin, that's right. He was the nicest, most gentle, most conversant, most accommodating person you can possibly imagine. And Rous was exactly the opposite of all those things. And they worked together for, like 10 years. So
should we now just go over what happened to RSV in the next 20 years? Because this is very cool.
You bet.
It's something we will come back to in Virology 101 when we talk about transformation. But I find it really neat that Howard Temin really made the next big step.
Another Rockefeller guy.
No, he wasn't a Rockefeller guy.
He was there.
No. He was a postdoc at Caltech.
Yeah, and then where?
And then he got a job at Wisconsin.
Right.
At the McArdle?
Yeah, yeah, in fact. He
never worked at Rockefeller?
No, he never did.
So. I'd be darned. Sorry about that.
So he went to Caltech to work on fruit flies. Restless and imaginative, Temin soon grew bored with fruit flies. So he switched to study Rous sarcoma virus in Renato Dubecco's lab. And he basically devised an assay for measuring cell transformation caused by Rous sarcoma virus. So Temin basically devised an assay for measuring transformation of cells. He used cells in culture and showed that if you infected with the virus, you got little foci of cells piling up which were transformed. That is, their growth properties were altered by the virus, which is one of the steps on the way to being a tumor.
They lost contact inhibition.
There's contact inhibition. They pile up. They make little foci.
Yep.
Right? Yep. And from that, he figured out that this... By now, we knew that this virus had an RNA genome. Right?
What year are we talking?
Now we're in the 60s.
Yeah, 1961. They figured out that it had an RNA genome. I caught that from Robin Weiss's review. It caught my attention because that was done by Lionel Crawford and his wife Elizabeth.
So that's important because then Temin now says, look, I infect cells and they become transformed and they stay transformed. And he said it's got to be that the genetic information of this virus is now in the cell. But how can it be if it has an RNA genome?
Right.
So he made this leap of faith. He said, I love the way Mukherjee puts it. Temin made a leap of faith. If the data did not fit the dogma, then the dogma needed to be changed. That's great. What a great writer.
That is so important.
It is.
Just as a fundamental principle.
Yep.
And you mean in general, we should be willing to change the dogma?
If the data don't fit the dogma, you got to change the dogma.
So as long as the data,
that's the scientific method, right?
Yeah, as long as the data are good. We know sometimes the data are not good.
Yes, the data have to. Extraordinary claims demand extraordinary evidence, which Temin eventually got. And
he said this has got to be. There has to be an enzyme in this virus that copies the RNA into DNA.
A reverse
transcriptase
of fortune.
So he found it, and at the same time, so did David Baltimore. And then these two guys got the Nobel Prize for that.
Right.
And that was by then 1976, something like that. Let's see. It's 76, 77. Someone has to correct me because I don't see it here and I don't want to interrupt this riveting story. So we got reverse transcriptase, which means Rous can convert itself from RNA to DNA.
Right. In fact, Vincent, there are 7 different ways that a virus can actually replicate inside of a cell.
Reverse transcriptase was discovered in 1970.
And the Nobel Prize?
The Nobel Prize was 1975.
Wow.
So I went to David's lab in 1979. It was just 4 years later. So I was pretty lucky to get in there. That was pretty cool.
Luck had nothing to do with it.
Yeah, it's lucky. Yeah.
I think skill.
So then the question is, why does this virus do this? Why does it transform cells? Why does it cause cancer?
Right. What benefit is there for a virus to do this?
Are these why questions?
Oh, that's right. I guess they didn't listen to us back then. How did these guys figure out, how did they find Sark? That's what we got to get to now.
Right. So the question was, what's going on here with this transformation thing? How do they transform cells? Or you could even ask, for what purpose, if you wanted to go there.
Not why.
Not why.
Okay.
We don't ask why questions.
Got it.
Right. So we're asking what's the evolutionary advantage to the virus in doing this?
Right,
I'm not convinced there is one.
So all right. So this is it. They made mutants which did not transform cells. They identified viral mutants that didn't transform, and they turned out to be in a gene. Are you guys there?
Yeah.
Right?
We're listening with rapt attention.
It was in a gene. And that gene they called SARC, SRC, short for sarcoma. Because they said, this is the gene. Although that was quite a leap, going from transformation to sarcoma. I guess they did test for the ability of these mutants to make tumors, and they did not. So they called this gene SARC. So that's actually kind of neat because you make a mutation in a gene. And remember, back then we didn't have any sequence, so you just call it the gene after the phenotype, in this case, sarcoma.
Right.
And do we know here who did that? It's all
that was done by Steve Martin, Peter Vogt, and Peter Duesberg.
Okay.
Not that Steve Martin.
Not the same Steve Martin, not the comedian, no. And this was all done in California, which Mukherjee says in the 1970s was the hotbed of cancer virus research. And they identified a SARC gene within Rous sarcoma virus.
Now, the virus still grew, right?
Virus grew just fine with this mutation, which suggests
it no longer transformed the cells.
Yeah, the transformation wasn't so important for virus growth, right?
Right.
And so they called this an oncogene, this SARC. And this is about the time when Temin said he made this hypothesis called the what was it called? Proto-oncogene hypothesis. Mm-hmm. He said this is a cellular gene. It started in a cell and somehow it got into this virus. And that's what's going on here. It didn't originate in the virus. The virus picked it up from a cell. So that's really an important twist in this whole history. Right. The idea that these oncogenes come from cellular genes. And so it was left to some people to find the SARC gene in cells, and that's Mike Bishop and Harold Varmus. They did that in the 80s. And they identified a cellular SARC, and they showed, most importantly, that it was different from the viral SARC that was carried by Rous.
Not a lot different, just a couple of point mutations, or even one, I think.
Right.
Yeah, one amino acid change, and the idea was in a cancer that one amino acid change in SARC was what was responsible for initiating the cancer, and a virus picked it up accidentally was the idea, and then could bring it to other tissues and other animals and transmit the tumor.
Right, and the previous work was essential in understanding how that could work, because once you understand the reverse transcription process and the virus integrating into the genome, then it makes sense that you could actually end up with a host gene mutated in the virus.
Right. So we have 3 Nobel Prizes coming from Rous sarcoma virus. Yeah. Rous's, Baltimore and Temin, Bishop and Varmus. I think it's amazing, actually. And when did Rous die, Dickson?
I think 2 or 3 years after I got to Rockefeller.
Okay, so he missed the SARC and all of that stuff.
Oh, sure.
Yeah, it's too bad. He would have liked to see it because he was poo-pooing, the cellular somatic mutation. But in fact...
his own virus did it.
That's it. The virus picked up a mutated cellular gene. Yeah, a somatic mutated gene.
He died in 1970 at the age of 90.
70, that's right. That's right. That's the year I left Rockefeller.
Let me read this last summary of Mukherjee. So one of the unfortunate casualties of Rous sarcoma virus was Saul Spiegelman, who was a cancer virologist who worked here, actually, and who interviewed me when I came looking for a job here. Huh. And shortly after that, he died of pancreatic cancer.
Really?
But upon hearing that EBV was upon hearing about SARC being a normal cellular gene gone slightly awry, he said, all human cancers have to have viruses like this. And he looked and he looked, and he found evidence all over, but it was all wrong. He never found anything. So Mukherjee writes, Nature, Rous wrote in 1966, sometimes seems possessed of a sardonic humor, and the final lesson of Rous sarcoma virus had been its most sardonic by far. For nearly 6 decades, the Rous virus had seduced biologists, Spiegelman most sadly among them, down a false path. Yet, the false path had ultimately circled back to the right destination, from viral SARC towards cellular SARC, and to the notion of internal proto-oncogenes sitting omnipresently in the normal cell's genome. So that really summarizes it. Yeah. When he discovered, people thought, viruses cause cancer. And then no one found any human viruses, but then they figured out how SARC worked, and now we understand that there aren't many human cancer viruses, right? Pat Moore said there are 7.
Right, but if you add it all up, I think what they say is that about 20% of human cancers, I think that was his number.
Yeah,
I think,
are caused by viruses
or something like that, are caused by viruses. So in terms of prevalence, it's actually pretty significant.
They're caused by some kind of infection because H. pylori is in there.
Right.
Schistosomes, right?
Yeah, but those are co-carcinogens.
That's fine. That's okay.
Something else has to be there too.
So this whole way that Rous sarcoma virus causes cancer, by picking up a mutated cellular oncogene and then passing it on, it's an accident. The virus doesn't really need to do it.
Right.
There's no evolutionary selection. The only selection is that this virus pops in and out of the genome, and when it pops out, it sometimes can accidentally take an oncogene with it.
Yeah.
So the answer to the why question we were asking earlier is it's an accident.
Yeah, it is.
Right. So this thing starts out at some point in the history probably as just a regular avian retrovirus, like avian leukosis virus or something.
Exactly.
That accidentally picks up this gene and becomes oncogenic in the process. One of the things that fascinates me is trying to understand, and I don't think we can understand this, when, during the course of Rous's work, this happened. Did the original sarcoma actually, did that have a sarcoma virus in it? Or was this somehow generated during his experiments passaging the tumor? Because after all, it did become more virulent as he passaged it. And I don't think we'll ever know the answer to that, but it's an interesting question.
Well, all the sections are still at Rockefeller.
Good point.
All you'd have to do is try to rescue it from the sections.
Yes,
very interesting. The technology exists now to figure that out.
That's really good.
Hey,
yes. Whoa. Definitely.
That's a very interesting.
Because
I'm feeling better already.
Here's the thing, Dickson.
Yes.
All the chickens in the world are infected
right.
by avian leukosis virus.
But they don't all get it.
Yeah, only about 3% develop leukemia. But as the chickens age, many of them developed a variety of other tumors, including sarcomas. And what happens is this avian leukosis virus picks up oncogenes, and that makes a Rous-like virus.
Right, but chickens usually don't live long enough for that to happen.
That's right, you kill them, right.
3 or 4 months and down the road, that's on your table.
That's now, right. is the viral... is there actually going to be recoverable DNA in those sections?
Yeah, sure.
Well, that's how
the virus is there.
That's how they reconstructed.
They got the 1918 flu.
Oh, that's true. The 1918 flu.
And there's a huge record. I mean, it's an enormously accurate and well kept historical record.
That would be fascinating to deep sequence those specimens.
Yeah.
Somebody should do that.
It needs to be done because this is a very interesting question.
I'm hearing an RL1. Somebody write a grant.
If someone at Rock has to.
Oh, I must mention also they've given out Nobel Prizes for the causes of cancer that had nothing to do with cancer. There was one called Spirocerca lupi.
That's right.
Which is a worm that lives in a cyst inside the esophagus of dogs. And because it develops a tumor that then metastasizes, they thought they had identified another cause of cancer, and that was back in the 19-teens or 20s, as I recall. And, of course, it was true for that particular organism in those particular dogs, but it didn't have any general application to anything else.
So one of the things that interests me about all of this history is this tendency of people to become single-minded about stuff.
Oh,
at first it was all
personalities.
Chemicals caused
Sure.
somatic mutation hypothesis.
Yep.
Then it was all viruses, okay? Then no, it's not viruses. Right. And in fact, all along, it's been all of the above, okay?
Right.
Well, and it's an important lesson that I try and keep in mind when I'm tracking things in the laboratory. You have a tendency to look for some single explanation for something that's going on, and lots of times there are multiple explanations for something that, looks like the same phenomenon.
Yeah.
When your favorite tool is a hammer, you see all problems as nails.
Right.
Hey, Rich, don't you think that's
true.
in part because of the publish-or-perish nature of science?
Not then.
Well,
but today it exists still.
Yeah,
I think it's just human nature.
Yeah.
Yeah,
I
think there's a lot of human nature.
It's comforting to have your beliefs validated.
Right.
Of course it is. And the other thing is, of course, up until the late 1950s, early 1960s, Rockefeller never encouraged anybody to apply for grants. That was all funded through the Rockefeller Fund. So it wasn't about the money. It wasn't about the size of your lab. It was a shark tank from start to finish for domination of the biological literature. And, they were raging arguments as to whether proteins or DNA or RNA was the genetic material until, of course, it was resolved. Mm-hmm. And there were huge laboratories devoted to all three of those areas. And, of course, when the winner turns up, everybody else sort of goes home with their tail between their legs. So
we will revisit transformation again in another Virology 101, because that in itself is a wonderful story, which we've only touched on here with Sark.
Good stuff.
But there are a couple of good articles about Rous, which we will put links to. One is by Robin Weiss and Peter Vogt. Vogt, of course, one of those involved in identifying Sark initially.
Yes.
Called 100 Years of Rous Sarcoma Virus. And Dickson, it was published in... What do you think?
1912.
Now, what journal?
Oh, the Journal of Experimental Medicine, of course.
And then there's another review article by Harry Rubin, who also, who apparently was involved in early assays of Rous sarcoma virus transformation. He's written a review article as well. The early history of tumor virology, Rous, Riff, and Raff.
Would you allow me to just interject one more thought?
No, I'm sorry.
Oh, please. Now that I'm feeling better, come on.
You
my interferon kicked in, guys. It works. So there are two other parasites that induce cancers. One is called Clonorchus sinensis. Mm-hmm. It's a liver fluke that lives in the common bile duct, basically. And it secretes really potent carcinogens, a chemical carcinogen to be sure. And the other one is called Opastorchus viverrini. They're related to each other. And they're found all throughout Asia and South Asia. And they're transmitted by the ingestion of raw or undercooked fish. And liver cancer is very, very prevalent in those regions, and also from hepatitis B. But in this case, 2 parasites are really implicated in carcinogenesis as well. They secrete weird things like high levels of proline, which has a proliferative effect on the epithelial cells that line the common duct. Then they go down and eat them. When they migrate back up the duct, they secrete more proline. The cells
farming.
They're farming, exactly, vertically.
By the way,
they're quite remarkable
Dickson,
animals. Yes.
Speaking of causing cancer,
Please.
So there are 7 human cancer-causing viruses, but as far as I know,
Yes.
correct me, gentlemen, if I'm wrong, none of them work by passing an oncogene from a cell, as does Rous. They work by other mechanisms.
Sure.
Okay,
Right.
Everybody okay with that?
Yeah, I'm okay with that.
I think I'm okay with that.
So this mechanism that
definitely
we've
okay with
gone
that.
through was important in just elucidating one way that a virus
Right.
can cause cancer. There are many other ways.
Sure.
Okay.
But this mechanism also turned us on to oncogenes.
It
There
did.
you go.
Okay. Which are involved in other mechanisms as well, but we'll get to that another time. For now, though, let's read some email.
Yes.
Rich, can you take the first one, please?
Sure. Michael of Germlines, a podcast, writes, Dear TWiV,
Yeah.
there seems to be now a competition out there for addressing us.
I love it.
Yes.
I love it.
We got to collate all of these into one place.
TWiV, I like it.
Yeah, okay.
I greatly appreciated your discussion this week regarding B-cell immunology. That was a couple episodes ago.
Yeah.
Fascinating stuff. I was also pleased with your responses to one of the listener letters you read at the end of the episode. In particular, you guys did a fantastic job dissecting the misrepresented data that a blogger used to promote the fallacious claim that H1N1 vaccine and the HPV vaccines are associated with increases in miscarriages.
Right.
I went to read that blog post after listening, and I was horrified at this bunk, as I am with all the bunk that is being propagated, because it is so dangerous when people believe this nonsense.
Yep.
I am an epidemiologist, and I have to say, Rich, you hit the nail right on the head by pointing out the lack of a denominator in the estimates on which these so-called associations are based. Unfortunately, you find people doing this kind of thing all over the place. Mainly, I just wanted to write and say thank you for giving a clear description of how this kind of presentation of data is wrong, and to tell you that your episode inspired me to record an episode for my own podcast, Germlines, as a basic introduction to vaccines,
Cool.
what they are, how they work, and what the research process is that they must all undergo to prove safety and efficacy. It is a sort of a vaccine, basic vaccines 101. I recorded it this week, and I'll be posting it sometime in the next 2 weeks. So thanks for the inspiration. That's terrific.
Wow.
I just want to say that Michael will be on a TWiV early in January. We're going to do an epidemiology 101.
Excellent.
That would be
Excellent.
cool.
No, that's great.
At any rate, I want to point out that I can't really take credit for the lack of a denominator thing in that discussion, because I really want to make this point, because when I first looked at that blog, I was confused about how to look at it, and contacted a resource at the Centers for Disease Control, who's a vaccine safety expert, who described to me how the vaccine adverse effects, VAERS, how that resource works, and pointed out to me another resource that does have a denominator, and pointed out to me that there wasn't a denominator. Okay, the point is, I didn't even know quite how to look at this either, so I asked somebody, okay, who was reliable, okay? And he actually sent us a couple of papers that perhaps we can go over in the future that talk more specifically about how those databases work.
Great. All right, Alan, may you take the next one?
Yes, I believe I can. Junio writes, Hello, TWiV crew. It is fascinating to learn how viruses make the cell machinery do things that the machinery is designed to do for the virus's benefit. But I'm wondering how much of this mechanism how much of the mechanism explained in this episode, and I guess this is the RNA synthesis, is specific to synthesizing virus RNA from virus DNA as opposed to normal cell machinery being abused to work on non-host DNA. For example, splicing was mentioned early in the episode, but does it also happen when synthesizing RNA from host DNA? I assume the next step after virus DNA is transcribed to RNA is for the RNA to be translated to protein. I might be getting ahead of myself, but will we see Virology 101 translation episode later? And if so, I wonder how much of of what happens at the ribosome to produce viral protein specific to the virus replication cycle, as opposed to being a normal cell machinery that happens to be working on viral stuff. In other words, does translation in the Virology 101 class make sense, or is it just Biology 101? Okay, so in order, splicing does indeed happen in the regular transcription, the regular process of RNA synthesis. So the DNA gets transcribed into RNA as a full length in the normal cellular genes. And in fact, the structure of the normal cellular genes, which have these coding and non-coding regions, is the reason why you have splicing. So it splices the RNA into the final messenger RNA. And the reason we only briefly touched on it is because it's a whole other subject. Really, it's a whole aspect of the biology of RNA. But yes, that is a phenomenon that is natural to the cell, that occurs all the time. And what's going on with viruses is that they're sneaking their DNA in and getting it transcribed, and they just have to deal with the machinery as it is.
I mean, he also gets at... I'm sorry, Rich, go ahead.
Basically, all of those things we talked about are basic cellular processes. I think of a virus infection as just taking a new set of instructions, a new piece of, in particular with DNA viruses, a new piece of DNA and inserting it into an ongoing process. Now, obviously, the viruses tweak that process as well, but they're fundamentally using a lot of the cellular processes. And in fact, viruses historically have been used to understand the cellular processes because they're easier to study.
Right.
So he asked how much of the mechanism is, virus. How much is it virus?
It depends on the virus,
yeah,
too.
It really does.
Exactly.
As we mentioned, you have the very, very small viruses that rely on the cell's machinery for everything, and then you have very large viruses that pretty much pack all their own stuff, and they mostly shut down the cell's machinery.
Right.
And in between, you have a whole spectrum of other options.
So is there a translation, TWiV, translation Virology 101, Vincent?
Yeah, in fact, viruses do tweak the translation apparatus.
Yes.
Perhaps not as much, as far as we know, as much as transcription, but we do have a chapter in the virology textbook.
Great. If there's a chapter in the virology text, then there's a Virology 101. We can do a splicing one and a translation one.
But I don't think there would be anything wrong with Biology 101, right?
Mm.
Well,
that's
there's
another podcast.
As we've said, there's enough overlap in some cases so that they're really indistinguishable.
Yeah,
right. This is abnormal Biology 101.
Young Frankenstein?
Yeah.
Yes.
Josh writes, Dear TWiV Doctors, regarding the SIDRAP release about the influenza virus that will kill us all, and the letter from Thomas on TWiV 162, I have something else relevant to the discussion. It's a book by Nobel Prize-winning professor of physics Robert Laughlin entitled The Crime of Reason, where he discusses scientific knowledge that is being hidden, privatized, and classified. He discusses how nuclear weapons information was and is hidden. Ironically, he can't tell us what it is because he has a Q security clearance, and how that model is being extended to the life sciences in the name of bioterrorism. It's not a great book. It's short, and he's not the best writer, but the topics he raises are interesting, and his insider perspective is unique. A better writer than him with a background in the life sciences needs to take up the subject and explore it in more depth. Alan Dove, I'm looking at you. Nice.
Don't you dare go offline again.
A ringing endorsement, but it is
Very nice of you to say that.
It is it sounds like a really relevant book to this discussion that we've been having about this. His radio interviews have been interesting. Here's some links. He provides a few links to his interviews and his book, which we will post. Anyone aware of Robert Laughlin?
Not me.
No.
This is a new one.
Sounds like a good one, and certainly in light of the H5N1 discussion.
Yes.
All right. Rich.
Johan writes, just found your podcast searching for TWiV. I am a biochemist and really fell in love with your podcast. One pet peeve, though. Please make more video episodes. And he links to one of them, says, is so empty. It just makes it more pleasing to see the person that talks. Although I really enjoyed the video of the last episode about transcription, even though there's no face. Thanks, Johan.
Johan's from Sweden.
Okay.
Yeah, just we are planning to do more video. It just
we need to do three-way video. Actually, what we need is that grant that we're going to write to support a technician and some equipment to help us do that. Right?
Roger. Yeah, I'm writing it next week, actually.
All right.
It's next on my list. Alan.
All right, James sends us a link, and this is a link to a news story. And it says, I am most interested in this statement. One smallpox expert, Dr. D.A. Henderson of the University of Pittsburgh Biosecurity Center, said STS-246 has not been proven to work. Do you all have any opinions on either the STS-246, whether it's effective against smallpox and or the allegations? And this is a news story about the awarding of a contract, a large contract to a particular company that makes this drug STS-246, or
it's actually ST-246.
ST-246 is the actual name. CNN got it wrong.
STS is the shuttle number, right?
Right. Right. So the Sega Sega Technologies won this no bid contract worth $433 million to produce
million. I'm sorry.
Yeah, million to produce 1.7 million doses of this this drug. It's an antiviral to use against smallpox, and the idea is to have this on hand in case smallpox gets out and we need the drug. And then it puts that in the context of the overall biodefense investment. But the controversy is that there's the controlling shareholder of Sega Technologies is this fellow Ron Perlman, who is well known for a lot of things, but one of them is that he is a very big supporter of the Democratic Party. So there's this allegation that this was a political move, that the Obama administration gave the contract to this company because of that support.
I asked Grant McFadden about this, and he's got his opinion here if you want to read it.
I think it's worth reading.
Yeah, I'll go through this. So Grant responds, the drug in question is called ST-246. It was developed by Sega to target the viral F13 protein, which is highly conserved in all orthopox viruses, and block the release of extracellular virus at the end of the replication cycle. ST-246 is highly effective against every orthopox virus disease model tested to date, including monkeypox virus in non-human primates. It's also very safe in human clinical trials that measure safety or the ability to be used in conjunction with vaccinia vaccination, so it doesn't hurt the vaccine. It's also been safely used on an emergency basis. All the patients recovered from their disease and survived in 3 clinical cases of runaway vaccinia infection, which is a possible complication of the vaccine. The only real still unproven aspect about this drug is how well it will work against variola in people. ST-246 works very well against variola in cultured cells, but unfortunately there's no good animal model to test for the ability of this drug to treat the disease caused by this virus in humans. The closest available animal model is to inject high doses of variola intravenously into non-human primates, which avoids the early phase, prodromal phase of the natural disease and proceeds quickly to the end stage, which is a high viremic phase, when it kills the monkeys very quickly in a disease that doesn't mimic smallpox well at all. Since the drug's designed to stop the amplification and spread of the virus, it blocks the egress of the extracellular form of the virus, It's predicted to be less effective if given only at the time of end-stage disease. But even so, ST-246 still reduces the skin lesion count in Varroa-infected non-human primates, and that's exactly what the FDA is struggling with right now in terms of whether to license the drug as a therapeutic measure to treat smallpox. The current status of animal models using Variola virus are not good enough to unambiguously answer the question. But the overwhelming evidence is that ST-246, as well as a competing drug called CMX-001, are safe and efficacious against all orthopoxvirus diseases. But the evidence relating to how well it will work against variola in humans is still indirect because of the inherent limitations of the available animal models. And apparently the FDA met, I guess, last week. Discuss that.
Do we know the outcome of that, Rich?
I've talked with both Dick Moyer and Grant McFadden were there, and I've talked with them, and I don't think that there's any final outcome, but the impression is that they're moving closer towards coming to some sort of resolution on this. Okay, that there's been so much involved in this, and ST-246 has been proven in so many different animal models that there really can't be much question in any reasonable person's mind that it's going to be efficacious. You can't do anything more to test it. And there can't be any doubt in any reasonable person's mind that it's as good as you're going to do in a drug in humans. So it remains to be seen, but seem to be moving in the right direction on getting it approved.
What do you think, Rich? Should it be approved or not?
I think it should. Okay, so actually, I have a lot to say about this. But
you can talk. Go ahead.
So we should put a link to this paper that's a review by Robert Jordan as first author, Dennis Ruby as the last author, that talks about the development of ST-246. I mean, first of all, it's important to point out that this has been going on for almost 10 years now. That on the heels of the terrorist attacks and the anthrax incident, the biodefense effort in the country was ramped up, and following on recommendations from the World Health Organization recommended for all Class A agents, and at the top of that list is smallpox, that there be vaccine and 2 antiviral drugs. And basically the U.S. policy piggybacked on that and said, yes, we need a vaccine, and it's not really clear if they specified exactly how many drugs, but certainly antiviral drugs in addition to that. And so a small company called ViroPharma, I think I've talked about this before, in Pennsylvania, started up some years ago by Mark Collett, took up this charge and developed a high-throughput assay using cowpox infections in culture to screen a bank of drugs for antipox viral activity. Robert Jordan was the lead scientist on that, and he came up with this drug. They did most of I think they actually, the ST-246 is after some developmental chemistry on the original hit, and that was all done at ViroPharma, and then ViroPharma went belly up. And Sega, which was at the time quite a small company that had been around doing in various different formats, doing stuff for a period of time, bought the rights to ST-246 and acquired the remnants of the NIH grant to keep doing the research. And in fact, Robert Jordan moved to Sega. And they have done an absolutely fabulous job over the years of developing this. It's a model for drug development, and it is as good as you could want for an antipox viral drug. So anybody who wants to criticize the notion that there should be an antiviral has to understand that this is what the government asked for, and the scientists delivered, and they delivered in style. So any questioning of whether or not this should have been done, they have to look back at the real history of this.
Well, and I think it's important to point out, too, that there's not a huge number of these compounds developed by a bunch of different companies. So the criticism in this particular CNN report is that it was a no-bid contract, but it's not like there are a dozen other companies that would have bid on this.
No, and this is where BARDA steps into this. We had a whole episode on BARDA some time ago when we talked to Ed Niles. There's going to be no big pharma that wants to develop this because there's a single buy and a single buyer, and it's not something that's going to be marketable on a regular basis. So if the government wants this and they want to make it and they want to develop it and stockpile it, they got to put in the investment, and that's done basically through this government agency called BARDA. Now, there's been 2 drugs developed, ST246 and CMX-001, and in fact, they work by entirely different mechanisms. ST246 inhibits spread. CMX-001 is a nucleoside analog that inhibits DNA replication. So they would actually be great in competition, but I think the thinking now is apparently that we don't want to spend the money to develop and stockpile both drugs, that we're going to go with one, at least for the time being. And the judgment was made, and my understanding is this has nothing to do with politics. And I certainly, if I had to choose between those two, would choose ST246. They're both good drugs. They both work. ST246 has been, you can't test CMX-001 in non-human primates because monkeys metabolize this stuff. Okay, whereas ST246 has been proven effective in a non-human primate model. So the government chose what they felt was the best drug, period.
Right, and there's a pretty good basis for that, which is that's the drug we have the best evidence on. No offense to this other CMX-001, it's just that it can't be tested as thoroughly, as you pointed out.
Right. Why is Henderson saying this, that it has not been proven to work? Did they take him out of context, or is he just being difficult?
I don't know. I can't really speak to that.
I have a hard time explaining a lot of his statements lately.
Right.
Okay.
But it's not an accurate statement. Okay. That statement that it has not been proven to work. Now, if you say it's not been proven to work in humans, that's true, and I'm looking for volunteers
against smallpox, but it does work against vaccinia.
Well, there's anecdotal reports, right?
No, you can't do a clinical trial.
No, you can't do a clinical trial in humans. That's the whole problem.
Yeah.
But in terms of animal models, and there's a wonderful paper and table in this review showing all of the different models that it's been tested in. Let me find it here. I lost it. Okay, but at any rate, a whole bunch of different a whole bunch of different animal models, including both mice. Oh, here it is. Sorry. Vaccinia in mice, cowpox in mice, ectromelia, that's mousepox in mice, monkeypox in squirrels, rabbitpox in rabbits, monkeypox in monkeys, variola, that's smallpox in monkeys. It's been tested in all those models and shown to be, by some measures, efficacious. So it's going to be hard to do much better than that.
Right. That's what's available. I will say that none of them are humans, so you never know. But if you wanted smallpox drug, that's the best you're going to be able to do.
That's right.
And so,
And in terms of where a story like this comes from, my assumption is that the other company, Chimerix, may have had a hand in putting a bug in somebody's ear about, hey, this no-bid contract was awarded, because, of course, they didn't get it. And also, this is, it's quite obviously something that somebody can make political hay about.
Yeah.
So
now, in terms of the final price tag for this, that's above my pay grade. Okay, somebody else can haggle over that.
Right. I think we'd need a you'd have to do a risk-benefit analysis, but we don't actually know what the risk is.
Well, I'll tell you, I'm not on the NSABB, but this, I think, is one of the viruses that a bioterrorist would use, because we know it's got a high mortality rate and it's out there. So I think we ought to have a stockpile of vaccines and antivirals.
We're so close. Okay? You've been working on this sucker for 10 years. All right? At least. And it's good. And it's just a matter of producing clinical grade stuff and stocking it away.
Yeah.
Let's finish the job. We've invested a lot of money in this.
Yeah.
All right.
You want to complete the super collider down in Texas also?
Yeah, as a matter of fact.
And how about the Webb Space Telescope?
Yeah. Give me control of the budget. I'll be around.
Dickson, I think we need to spend a lot more money on science and technology.
Of course we do.
A lot more.
Which have more than paid for themselves many thousands of times over.
No question about it.
It's not that you're spending money on science, you're investing money in science.
Hear, hear.
Oh, boy. Let's move on to a few science picks of the week, Dickson.
Yes.
Well,
do you have a pick or you want to wait?
I'm going to pass on this one.
Okay.
No problem, Dickson. Rich.
Okay. In the spirit of the season, I'm going to choose a book called Log from the Sea of Cortez by John.
This is awesome.
So, a little background here. For those of you who don't know, John Steinbeck was a writer from the early 20th century who won the Nobel Prize in Literature for his writing. Most famous amongst those probably is The Grapes of Wrath, a novel about the Depression. He was a novelist. My favorite he wrote a number of novels, short stories, etc. My favorite of his books was, and this is relevant to this discussion, a book called Cannery Row.
Yes.
Which is just a fantastic book. As a matter of fact, it's relevant because there's a line in that book where these bums are on a little... It's about bums and prostitutes and this guy named Doc in the Cannery Row.
That's right, he's a marine biologist. That's
okay?
right. Nick Nolte.
And at one point, the bums are on some expedition to go collect frogs for Doc, and they run over a chicken, right? And they decide to cook the chicken, and they're sitting around the campfire, and one of them asks, how long do chickens live? And another one says, nobody knows.
That's right.
So, at any rate, Doc is relevant because in Cannery Row, because he is fashioned after a good friend of Steinbeck's named Ed Ricketts, who ran a biological supply company in Monterey and was a sort of a freelance marine biologist. He published stuff. He had his own collection, and to make a living, he had this laboratory on Cannery Row where he collected stuff and sent out, stuff like frogs to high schools and other samples.
Like wards.
Living.
Yeah.
So at one point in about 1940, Ricketts and Steinbeck in a rented boat went and did a collecting expedition in the Sea of Cortez, which is in the Gulf of California.
It's in Baja, right?
And they kept a log, and in fact, part of this is actually, although he doesn't get credit for it in the current volume, part of it is actually written by Ed Ricketts. And in fact, in reviewing this, I read this book in graduate school, so I haven't read it for 40 years. But I need to go back and read it again. Apparently, there's a chapter in this written primarily by Ed Ricketts on teleology, okay?
Right.
All about the why question. So I need to go back and read it again, and we'll do that again. However, my favorite passage from this, I want to read for you.
Mm-hmm. Go ahead.
Our own interest lay in relationships of animal to animal. If one observes in this relational sense, it seems apparent that species are only commas in a sentence, that each species is at once the point and the base of a pyramid, and that all life is relational to the point where an Einsteinian relativity seems to emerge. And then not only the meaning but the feeling about species grows misty. One merges into another, groups melt into ecological groups until the time when what we know as life meets and enters what we think of as non-life. Barnacle and rock, rock and earth, earth and tree, tree and rain and air. And the units nestle into the whole and are inseparable from it. Then one can come back to the microscope and the tide pool and the aquarium, but the little animals are found to be changed, no longer set apart and alone. And it is a strange thing that most of the feeling we call religious, most of the mystical outcrying, which is one of the most prized and used and desired reactions of our species, is really the understanding and the attempt to say that man is related to the whole thing, related inextricably to all reality, known and unknowable. This is a simple thing to say, but the profound feeling of it made a Jesus, a St. Augustine, a St. Francis, a Roger Bacon, a Charles Darwin, and an Einstein. Each of them in his own tempo and with his own voice discovered and reaffirmed with astonishment the knowledge that all things are one thing and that one thing is all things. Plankton, a shimmering phosphorescence on the sea, and the spinning planets and an expanding universe, all bound together by the elastic string of time. It is advisable to look from the tide pool to the stars and then back to the tide pool again.
Nice.
That is poetic.
Excellent.
Steinbeck, man.
He's a very powerful writer.
Yeah, I also read that a number of years ago, and not as long ago as Rich did, but
You were haploid.
I should go back to it,
right.
definitely.
I have to read it. I never did.
Yeah.
Yeah,
Will
great
do.
book.
Thank you.
Maybe
Rick.
I can tag on to that one, since I had nothing. Travels with Charlie is another great book.
Yes.
I would, anybody who's interested, it's almost the first thing you ought to do is read Cannery Row.
Oh, that's
it's
true.
Short,
Right.
it's quick, it's just absolutely delightful book.
Right.
And in the front of the log from Sea of Cortez, he has a section called About Ed Ricketts, which is basically, it's about Ed Ricketts. And it is terrific as well.
Wonderful. Thank you, Rich.
Yep.
Alan.
Well, that actually segues pretty well into mine. It's another of these astonishing time-lapse videos from NASA. This one is a comet rise.
Oh.
So this is it's shot from the International Space Station, which is orbiting the Earth very fast. And one of the astronauts shot this time-lapse sequence that shows the view as they're crossing the dark side of the Earth, the side away from the sun, and you see the glowing sunrise, and you also watch the rise of Comet Lovejoy, which has just completed circling the sun, and to the surprise of pretty much everybody, it came out on the other side. Everybody thought that it was so close to the sun, it was supposed to burn up in the solar atmosphere and such. But it came back out on the other side, and in this video you see the comet rise, and then you see the sunrise, and it's an amazing view.
Yeah, it's very cool.
Nice.
Very cool. In addition, I like when you're on the dark side seeing the thunderstorms.
Oh,
yes.
That's fabulous.
And then you can see, I guess there's this layer, this sort of rim on the Earth that I guess is the atmosphere.
Yeah.
Oh, sure.
Okay.
That red color.
Amazing.
Yeah, very cool.
Yeah.
Yeah, it's very brief. It's only 30 seconds.
Yeah.
And then
there's a little commentary by the
guy up in the
mission
space
commander.
station.
Did you watch?
Which is cool because his mind is blown, right? And
Right,
Yes.
right, sure. Did you watch the comet actually go around the sun? They had a video of that. They showed what happened to the comet's tail.
I was on it, Dickson.
It went around the sun. You saw the tail get lost, and then they had another video of it coming out or the other side. So you can actually go to NASA's website. I go there all the time.
Yeah.
It's a fabulous thing. And, of course, they're cutting their budget, too.
Everyone, yeah. Thank you, Alan. My pick is for everybody who wants to buy themselves a little holiday gift. And it's pretty much the coolest iPhone case that you can get.
I would have to agree.
Did you make this?
Yeah, I took the logo and kind of tweaked it. And,
It's very cool.
Till it fit the phone. So
I think I'm going to have to get it.
I like it. My phone has it, and it's the TWiV logo.
Give it to your graduate students.
So I put a link to a little blog post where I put the original artwork up. So we don't make any money on this, but there's a company called Uncommon that lets you put any art on iPhone cases. And I'm sure you could find the same for an Android case. I just happened to have an iPhone and made this and thought those of you who have such devices, it'll work on the 4 and the 3GS, would like to do this. So the link is there, the image is there, and you just order it. I think it's about $39 for an iPhone case.
Looks cool, Vince.
And if anybody wants to let me know about Android equivalents, I'll be happy to modify the image for that as well and put that up. So That's a little self-serving for TWiV, but I thought it's a good way to advertise.
I think it's cool.
Sure.
I think it's awesome.
Everyone I've shown it to
cool.
so far thinks it's pretty neat.
It is.
Because it's TWiV, and you go in an airport with this, people are going to look at it and go, what is TWiV.tv? Let me go there. And that's what we want. And, of course, you loyal fans should have one. So there you go. That's my pick of this week. And we have a couple of listener picks, 3 of them today because it's the holiday. Mm-hmm. First one is from Ronnie, who writes, "Hello, Professor Vincent. First of all, thank you for your wonderful podcasts. I am a CFS sufferer and also a student in applied science, so I'm interested in many of the topics discussed for those 2 reasons and always learning new things. I think I just found a great listener pick of the week. If not, you can always use it as a Christmas card. This is how fungi celebrate Christmas." And there you go, Dickson. See? Right. Christmas fungus on a Petri dish. Complete with ornaments. It's great.
Maybe, I wonder if that almost ought to be the episode picture.
It's pretty nice.
It doesn't look like Rous, but it is awfully cool.
You could do that. It's fine with me. And we have 2 others. Oh, I'm sorry. Let me finish Ronnie's email. Also, I've been following your blog posts about the H5N1 news the last few days, and it was just on the Dutch news that a debate will be held about the research in Dutch Parliament after the holidays. Oh, no.
Just after the flu season ends.
Yes.
What's Parliament going to do? It was quite a long topic on the news. They also interviewed Professor Fouchier. If you want a full translation, let me know, and I'd be happy to give you one. So I said, sure, let's have it. Greetings and happy holidays, Ronnie. So thank you for that. And I have 2 other picks. One of them came from Kevin via Facebook. These 2 picks, by the way, are iPhone apps. They may also be available for Android, but I don't know. The first one is called EcoCYC. EcoCYC is a database of E. coli. It has been around as a website for a long time. It has all the genes, all the sequences, all the annotations, everything you would want to know about every one of the thousands of E. coli genes. And this is basically an app which is a front end to the database. You can search. It gives you a list of all the genes, and you can click on them and find out everything you would like to know. It's a mobile interaction for EcoCYC. It is so cool. This is just where these apps are really shining for science, I think.
I wonder if the E. coli on your phone can access the app.
That's right.
No, it goes to the human genome.
Oh, yes.
It's trying to find a weakness, of course.
And the second pick is from our friend Gabriel Victora, who was on TWiV just a few episodes ago. After the episode was over, he said, I have a pick I forgot to give you. And I said, I'll use it another time. And this is also an iPhone app. It's called ImGen. And this is also a front end to a database. And this explores gene expression in different immune cells of the mouse. So you put a gene name in, and then it gives you a choice of 6 different kinds of immune cells: B cells, myeloid cells, NK cells, T cells. And you click it, and you get an expression. It's called a heat map, which is an expression level of the genes that they're interested in all the different cell types. So if you're
you can find friends, family, and neighbors of these genes.
It's a little humanized, right?
Yes.
So anyway, this is also cool. This is another great example of using these phones to do cool things in science. So thanks, Kevin and Gabriel.
Great time to be alive, It's amazing. Just amazing.
And God bless Tiny Tim. I just thought I'd throw that in because 'tis the season.
Okay, is that from the Christmas Carol?
Yeah.
All right.
Yes.
By... Look at, he's hesitating.
Charlie Dickens.
Charlie.
All right, thanks for those listener picks, and that will do it for TWiV 163. You can find us on iTunes, of course, Zazune Marketplace, TWiV.tv, and over at Microbe World. We also have an app for your iPhone or Android. You can find that at microbeworld.org/app, or you can go to Stitcher Radio. It's another app where you can put all podcasts on, and they stream live. You don't have to download them, which is pretty cool. If you do use iTunes and you haven't done so, go over and leave a little review there or a star. That really helps us to stay prominent so more people discover us. And, of course, send us your questions and comments to TWiV at twiv.tv. Next week, we will be doing our 10 virology stories of 2011. So if you have any suggestions this week, send them in. Should be fun. Been quite a year for virology.
Definitely.
Don't you think? I think so.
Yes, absolutely.
It's going to be tough.
Every year is quite a year for virology. This has been good.
We're going to have a hard time limiting it to 10, I'm sure. Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
And, you're out there in the woods where the internet isn't great, but we hope that in the new year it will be better for you.
Why, thank you.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
I'm speechless once again.
What are you doing?
Goodness.
You looking at something else?
No, I'm looking at my grandson on
sure.
Facebook. That's what's happening.
Yes, of course.
I got to go home and see him.
You're amazed that you have a grandson.
I'm amazed that I've got. I'm just generally amazed. Okay, I'm in the spirit of the season here.
All right.
My mind is blown. Okay?
Here.
Good. Way to go.
Yep.
Have a
So you're
great
welcome.
have
It's
a great
always a
time.
pleasure.
Have a great time with the Sturpies.
Absolutely.
Very good. I'm Vincent. I'm sorry, Dickson.
It's okay, Vince. It's okay.
Oh, you want me to ignore you?
Do what you want. I would prefer you not to, of course.
Dickson is at verticalfarm.com, trichinella.org, and medicalecology.com. Thank you, Dickson.
You're welcome. And guess who just endorsed my book?
Who did?
Ralph Nader.
Cool.
He had picks of the end of the year, and he said, this is one of those books.
Where did he do that? Does he have a website?
Yeah, he does.
Nice. Yeah. We'll put a link to it.
Yep.
Congratulations.
What kind of car did he recommend?
Not a Corvair. Anything but a Corvair. That's right.
That's good, Dickson. Well, we wish you many good books in 2012.
Thank you, and I'll try.
All right. Thanks, you. And I am Vincent Racaniello. You can find me at virology.ws. Whatever holiday you celebrate, have a great one, everyone.
Absolutely.
Thanks for listening. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.